Ladies and gentlemen and ladies, cats and dogs, children of all ages and everything in between. Welcome, welcome, welcome. You know who it is. It is the kid, Unc Just, the pizza freak, resonating at the pizza frequency. I love talking about pizza. I love making pizza, but mostly I love sharing pizza with people that I give a ship about. Yes, literally, breaking bread with people that I care about. And I have a sneaking suspicion that you too, my friends, also resonated said pizza frequency, or why else would you be here? And where is here? Glad you asked. Here we are. Science of Sourdough, our last one, I think, for 2023. This is part 7. Part 7, Science of Sourdough. You know the dealio. Noel is trying to get to the bottom of what is the sourdough, talking to our friends down in Sandeezy, Levin Foods, Cam and Jimmy. We got a really fun friend of ours, a new friend, Angela Oliverio. I hope I said that right. If I didn't, I told her she's in a good group. And she's gonna talk to us about the Global Sourdough Project, interpreting the figure. That's like me when, like, after I eat a bunch of pizza and I look at myself in the mirror. That's a joke. All right. You guys know the deal. Thanks so much for coming in, hanging out with us. I'm really excited to, we're going to have a PowerPoint presentation on the Zoom. We will not be bringing people up to panelists today. Ask everybody to just hang out and watch with us. Remember, if you are watching on your phone, go ahead and make it horizontal so you can see the PowerPoint a little bit better. The Science of Sourdough will be available on the Slow Rise YouTube channel very soon. But without any further to do, what I'm going to do is I'm going to bring Noel on. I don't have the blue bell because I'm on vacato in Hawaii for Thanksgiving. But Noel, if you're there, come on down, my brother. I see you sending me a bunch of things. Here we go. I got some links. I'll be putting some links into the chat. There he is. I'll be putting some links into the chat. You like my hat, Noel?
Justin, I love your hat.
Thank you. Feed me pizza. Feed me pizza. I just get right to it. Just get right to it. I don't want to.
You think you get to it, but I don't know if anybody but me understood what you said. Where are you?
I'm in Hawaii.
Hawaii.
Honolulu. Maybe if you're lucky, it's a little cold and rainy. We're having a blessing right now, a blessing from the gods. So grateful for the blessings always, always, always. I'm here visiting some friends for Thanksgiving. So
now, correct me if I'm wrong. You shipped yourself some pizza provisions in advance of your. Just out of curiosity, what did you ship to Hawaii that you can't possibly get there?
So it seems difficult. You know, I like my round Tupperware containers for my balls. So I had to ship those. I also shipped some flowers.
What kind of flour?
Some
Let us
Karen
know.
Springs. What I got?
Really?
I got some Trailblazer and some Glacier Peak.
Because they don't have Glacier Peak in all 50 states.
I don't know. There's not. I'm gonna go to Chef Zone after to see if I can get some whole milk mozz. So I'm looking forward to that.
At least they have a Chef Zone.
Yeah. Well, the problem is there's no King Arthur AP on the island right now. I don't know what's going on. I went to Whole Foods. I went to Food Lion. I went all these different places. I can't find it. So, but I'm excited to be here in Hawaii, but I'm also excited to be here with everybody on the Zoom. What are we doing today? What's going on? Last Science of Sourdough for 2023. Is that correct? Is that true?
Rumor has it. You know, it's we've been going strong for a couple months now, and I think we're tired. We're going to take a little break. We've all got a lot on our plates, working hard. I know Cameron and James are making microbes like crazy.
Right. Sequencing of the sourdough dough, right? I mean, that's a pretty big deal. That's a lot of work, right?
And I don't know. I don't know if it is. I mean, I'm told it is.
Okay.
It is.
Yeah, but you haven't actually seen them working. Is that what you're skeptical of how much effort they're putting in? What do you say?
I'm a little skeptical. I don't know about this whole science of sourdough thing.
These guys are just
science.
It's easy
really
down.
Is it really? People say pizza's a science, and I'm like, I'm definitely not a scientist.
Yeah, I don't know. They're taking it easy down Sandeezy, if you know what I
mean. Nah, not judging from the bags under Cameron and James's eyes.
Should we bring them on to say hey real quick? Or you want to?
I mean, I think that James is feeling shy tonight, but Cameron is feeling good. So Cameron, where's your blue bell, Justin? You didn't have that.
I wish I had it. I don't have it. I didn't bring it. That's one thing I left. My goodness. My goodness.
Next time we'll ship you a blue bell. There's Cameron. Hi, Cameron.
Hello.
Where does today find you, sir?
Finds me in San Diego doing well.
In San Diego doing well. Look at all your
Yeah.
toys in your office there.
Yeah.
I see
got
some DiNapoli tomatoes.
Yeah.
I see some flour. I think I see a sequencer in the background. Doesn't look like much. Looks like
Yeah.
an old printer.
Yeah. Yeah. They spared no expense in the design of the fancy sequencer there.
They got the
They slapped some LED lights on there and
Yeah,
called
there
it
you
a
go.
day.
I know,
You know?
right? I used to have a light bright that looked like it was more expensive than one of those.
I know. It fool you.
Yeah.
We don't tip off any break-ins. We need to,
Yeah. Keep it. It's incognito
Yeah, exactly.
sequencing. Yeah, there you
Yeah,
go.
they just think it's a printer and leave it there.
Nobody wants those anymore.
Excellent. All right, so I'm excited. Tell us a little bit about our guest and why you invited her.
Well,
and then we'll bring her on.
We got one of the most foremost experts in sourdough microbiology on today. Did one of the most exciting experiments that I think I've seen on sourdough, the 500 sourdough experiment, and I think has some really interesting points or maybe misconceptions about sourdough to talk about. And I'm very interested in her Acetobacter, which is a relatively low abundance microbe, meaning there's not a ton of it relative to like your lactobacillus and your yeast that we've talked about. But I think it's really important. And low abundance microbes are just as important as high abundance ones. You have one human in the rainforest, in the Amazon rainforest. They're not very abundant, but if that person has a chainsaw, they can make a big impact on that rainforest, right? So abundance does not equal importance. So I'm excited to learn today.
Awesome. I like that.
Great. Well, I just want to make sure everybody knows if you have questions, go ahead and put it in the Q&A. David Jacobs, want to give you a shout out. Yeah, no more office hours this year either. We're gonna take the rest of the year off. Is that correct?
We're saving our energy for 2024.
Yeah.
I think it's gonna be a big year.
Big things in Bente Quattro. Is that? Anyway.
Sure, Justin. Sure.
Okay. All right. And yeah, Dave. No, they Karen Springs. They stopped doing their free shipping on the big bags, so we can't. It's tough to get. It's tough to get the good flowers. But all right. What do you want to do? Let's bring her on.
Yeah, I mean, you don't have your big blue bell, so
The
without any
big
further
bell.
ado, see if you can pronounce her name. Come
Here
on.
it is. Angela Oliverio. There you go. Hey, welcome.
Hey guys.
How's it going?
Good.
Thanks so much for joining us. My goodness.
Absolutely. Happy to be here.
Yeah, this is going to be awesome. So where are you now?
I'm in Syracuse, New York.
Never heard of it.
Yeah, exactly. Yeah. Coming from Portland, Oregon and Boulder, Colorado, and everyone's like, wait, what? Why did you move to Syracuse? Certainly not for the pizza. That is true.
Some people might take offense.
Yeah, I know.
I know. I should be quiet because I still do get pizza here a lot. Don't want things.
Spill the tea, Angela. Spill the tea. All right. Well, I'm going to bounce off. Cam, why don't we turn it off and turn it over? So you guys enjoy the rest of the show, and we'll see you in a little bit.
Well. Here we are, finally. After so much ado, I'm super excited. I'm excited about your presentation. I'm excited because I don't know what any of this means. I mean, I read it over, but I don't understand 9/10 of what you and Cameron are talking about. So hopefully this will be an enlightening chat. I have a few softball questions just to get things going, if you don't mind, and then we'll kick things off. So did you grow up with an interest in either pizza or sourdough?
Pizza definitely came first.
Pizza came first. Do you have any early memories of eating pizza? Does it push a button or ring a bell or?
Been a long lover of pizza. I'll have to say in my youth it was probably like the bowling alley variety. It still hits a certain spot from time to time, right?
I understand.
But my taste might have sophisticated a little bit since then.
Or not.
Or now I just span the whole range, right?
Yeah.
And my son is now 18 months and a true pizza lover through and through. So it's come full circle, and I'm very, very happy to see that.
That's beautiful. And then how did you get into sourdough?
Yeah, sourdough. You know, I lived in a co-op in college, and it was just like super crunchy, nerdy. And so I kind of had to learn how to make bread to fit in there. It was actually on our chore wheel. We had a weekly chore wheel. And like making 10 loaves of bread for the co-op weekly was one of our chores. So that's kind of how I started doing sourdough. And then it became a research project, and then everything broke loose. So
Now, did you already know, like, as a Bachelor of Science, that you were going to go into the sciences?
oh, no way.
No way? Really?
No, I went around for a little while before.
Where did your journey begin?
Yeah, I was actually a philosophy major. And I moved back to Portland, Oregon after I graduated from undergrad. Worked there for a couple years, and then decided to get back into it and back into grad school. But Portland is definitely like the place to be, especially at that time, for all things fermented food. So
Yeah.
it's really an exciting place to be.
So you came to the science of sourdough from actually eating it, because I feel like there's a whole bunch of scientists who really couldn't care less about eating pizza, and they're more about the microbes. Is that a fair assessment?
Totally. Now I have to pretend that I'm more about the microbes, right? Make the pitch that I'm more about the microbes. And I do love the microbes, but yeah, it's definitely motivated by my own personal enjoyment as well.
And so apparently you got so good at it that the university where you work gave you your own lab.
Yes.
Is that true?
It's true. And I even pitched sourdough, and somehow they still said that they were going to go through with it. Yeah, I know.
Wow. It's very exciting.
Yeah, it's super fun.
Good. Well, thank you so much for joining us. We're excited to get going. I know that you have a pretty lengthy presentation, so I'm just going to remind you, please stop, because I'm going to have a lot of questions. I don't know much about the science of sourdough. I'm learning along with the rest of our attendees. So go slow and make sure to pause so I can get a few questions in here and there. All right?
Absolutely.
Thank you. Where shall we begin?
Here.
There we go.
Does that look good?
Looks beautiful.
Great. Okay, so as I mentioned, I have to, make my pitch a little bit broader than sourdough for the microbe world. So I just wanted to kind of share broadly, our research program at Syracuse. One of our main systems that we work in is Sourdough Starter. So we're going to talk about that in a minute for the rest of the day. But we also look at microbes that live in soil environments. And this is a picture of a geothermal spring system in New Zealand. So we're also really interested in sort of like extremophile microbes. And the pitch that we make now about sourdough is that it's actually a really useful tool to understand sort of basic biology and questions about microbes generally. So some of the big questions that our lab asks is, how do microbes interact with each other? And how do these interactions impact microbiome function? And so I just want to talk to you for 1 second about sort of natural system and what a microbiome looks like in a natural system. So I just want to give you a sort of scale of understanding, and soil microbes are actually some of the most diverse and abundant microbes on Earth. And if we just zoom in, this image that you're looking at right here, whoa, that's actually zooming in. Here, let me turn my laser pointer on. Okay, there we go. So this is the tip of a plant root. And if we zoom in on just this little section, so this is like the width of a human hair.
Wow.
And if we zoom in on just this little circle right here, you can actually start to see all of the individual microbes, right?
Wow.
And this is just to say that thousands to 10 to thousands of different microbial species can live within a couple tablespoons, which is just crazy. So soils are super, super diverse. They're really interesting to study, but also really challenging to study. And that's where sourdough starters come in. Noel,
Interesting.
anything before I jump into
Yes, yes. Well, you already jumped forward a slide. Go back one slide, please.
Oh, okay, let's go back.
Thank you. All right. So what am I looking at where it says approach and there's a triangle with a star in the middle?
Oh, yeah. So this is kind of like how our lab works. So we use experimental microbial systems, which would be, you know, sourdough starter, for example, right? So we actually are making synthetic starters in the lab, but then we do other things with those starters. So we sequence them, we look at their genomes, which is kind of like the blueprint for all of the different metabolic processes that they can do. So we use bioinformatics and different tools to look at their genomes. And then we try to combine these two things together and actually start to use data science and modeling approaches to predict sort of bigger questions, like can we take, you know, some type of information from the genome to predict what the pH of a starter will be, for example. So that kind of thing.
Gotcha. And there are people who are watching this who may have seen some of our Zooms before and maybe some who haven't. So when you talk about sequencing a starter, can you explain what that actually means?
Yeah. So sequencing is a really helpful way to actually understand what microbes are in a sample. And how we've traditionally done it is through culturing, which is also really useful. So if you take a swab of sourdough and you smear it on, you know, a plate that has nutrients on it, you can actually grow the microbes. And that's really, really useful, right? You can do lots of things with that culture. But you can also sequence the actual microbial cells, and that can tell you kind of like the taxonomic identity of the cell, so what species it is, and it can also give you other information about what the cell can do metabolically. So sequencing it is looking at the code of ATs, Gs, and Cs to get a sense of the microbe that's there.
Understood. And if I could bother you to go back yet one more slide to the very first. So what are we looking at there? What is that a photo of?
Yeah, so these are some of the microbes that we're actually growing in our lab. So when I say culturing, this is exactly what I mean. And we actually split our plates, our culturing plates, into quadrants because we can just get more use out of them that way. So here we're testing to see which microbes are persisting through sort of a time series test in sourdough. So you can see like here, this particular starter had it, but the other three didn't. So there's only growth in this one quadrant.
Got it.
Yeah.
Got it. Thank you for going back. I have lots of questions. There's a lot going on that maybe you take for granted, but I certainly do not.
It's good.
And so that I see a swath of earth right there, and I do see that you're measuring it. What is that meant to show?
So this is a classic soil depth profile. And honestly, the tape measure in this photo isn't really saying, I mean, it's showing the different horizons of the soil, but this is just to show that on really, really small scales, there's incredible microbial diversity. Yeah.
Understood. Thank you.
Yeah. And since soils are so crazy diverse and a lot of other microbial systems are also super diverse, our lab uses sourdough starter as a model system. And what that means is that we can actually take sourdough starter into the lab and manipulate it. And because it's has, you know, fewer species interacting with each other, it's much easier to sort of set up experiments and ask questions, and that we then can sort of scale up to these more complex systems.
So you're using the food that we feed our dough as something that's actually much more than that, huh?
Yeah, exactly. Yeah, so it's a really, really useful model system because it's pretty basic relative to a lot of other sort of microbially dominated systems.
Now you say that like it's a thing, but how long has that been like known, or how long has that been established? Is that like centuries old, decades old, or just a few years old, that idea?
Yeah, I'd say the idea of using sourdough starter as a model system in science is pretty new. It really started with Rachel Dutton, who is a scientist, and Ben Wolf, who's a collaborator, and they actually started using cheese as a model food system, and have since kind of expanded into a few different food systems. But the idea is that, fermented foods make so much sense when we're thinking about how to study microbes, because by design, we've manipulated them so that they're really easy to grow, right?
Understood.
And we've been doing that for thousands of years. Yeah.
Beautiful.
So this image is a zoom in of sourdough. So you can see these are yeast cells, and then these are lactic acid bacteria, and then these like rod cells are acetic acid bacteria. So those are kind of like the three main functional groups that you might find in sourdough starter. And I think that yeast and lactic acid bacteria are, really well known as key components of sourdough. And then one of the things that we've discovered is that acetic acid bacteria, when they're there, they're really important. So I'll tell you a little bit more about that too.
Love it. Let's do it.
Cool. Okay. So we started with this big question. And the big question is, what does sourdough starter diversity look like globally, right? And we really had no idea. And part of that is because even though there's a ton of interest industrially in, baker's yeast and lactic acid bacteria, almost all the research has focused on just a few very specific strains. And it's interesting because sourdough is, you mix flour with water, you put it out, and magically, like, the correct microbes are recruited, right? And that's true wherever you are in the world. But surprisingly, like, we had no idea, are the microbes over here the same as the microbes over here? And does it matter how much water I put in my starter over here or over here? So we really were interested in kind of understanding super, super basic, but not understood questions about the global diversity of sourdough starter microbes.
Got it. And when you say we, I see that this slide is the first slide that has your name along with Landis. Who's Landis, and how many people were working on this project?
Yes, Landis is the other co-first author. So we led the study together. And when I say we, I mean the broader group of scientists working on this project, and also the community scientists that were the folks that actually shipped in their sourdough starters to us, right? Because they were really important contributors as well. Obviously, we wouldn't have been able to do the study at all if they hadn't sent their sourdoughs. Yes, so I say we, and I mean broadly everyone who worked on the study.
Now, did you start out to do 500, or did you start out at 50 and then at 100 and then 250, and then you're like, oh my God, this is crazy, let's get 300, and you ended up at 5?
I don't even know what the original target was. And I say 500, but we actually landed on 560. And then we had to cut it off, right? Because it's funny. Initially, we were kind of like, oh, are we going to get enough starters from people? And will they actually, we'll be able to get enough to get interesting data? Definitely wasn't a problem. In fact, I was talking to my collaborator, and he said that their lab at Tufts is still getting sourdough starters. He's like, somewhere out there on the World Wide Web, there's a link that still says to ship to my lab's address. So it's a beautiful problem of
Yeah.
But obviously, part of the promise for the study is that, you'd get to then understand the actual microbes that are in your sourdough, which is really exciting. So there was a really important community science component this project, which was sharing out all of the data that often we bury in academic publications, right, with the actual people who wanted to know about the science of sourdough. So,
so I see that you have a lot of dots scattered through Europe.
Yep.
I might see something in Asia, definitely Australia, nothing in South America. I see 1 dot in Central America and a ton in North America.
Yes, good summary. And you're totally right. I know, and I've been saying global, and we actually purposely didn't say global in the paper because we're like, this isn't really global. So we called it intercontinental, but it's totally dominated by North America and Europe. Yeah.
Yeah. I mean, it looks like it's hard to tell. Like there might not even be any in Africa, or maybe just I see maybe 2 in Spain. So you think there's I mean, I'm sure they've been using sourdough starters in Africa for a long time, right? You just didn't get any in this particular.
Yeah. And I think it's partly like how we pitch it as sourdough starter, which has a really specific kind of meaning about a type of bread that we make versus a ton of other types of, fermented flour-based products that are more like regionally specific. And I would love to start getting into, a lot of different types of ferments and kind of the diversity there. So yeah, you're totally right. I think like the construct of sourdough starter and how we pitch it is something that is specific, and our sampling reflects that.
Understood. Thank you.
Okay. And then in addition to actually getting the sample of starters, which came in like little plastic baggies often, but some people sent a lot of their starter. We also got like letters from people telling us about, the history of their starter, and wanting to make sure that we really understood the sort of background, and including all of these sort of rich details that we don't usually get to think about as scientists. So I think that was really fun. We even got whole loaves of bread shipped with the starters, which was not, requested. But people were so excited to show off their starters. And I'll say, like, we started this project in 2016, maybe 2017, and then when we had the data, we were analyzing it. It was during COVID. And so then there was just this boom in interest in sourdough. So that was really fun.
Love it. These are great.
Okay. And then in addition to getting the starters, we also asked everyone who sent their sample to fill out a survey and to tell us about their starter, at least in a somewhat controlled way. So we asked about, what type of flour they fed their starter with. We asked about how they stored their starter, how frequently the starter was refreshed, how long their starter had been maintained for, and if there was a source to their starter, like if they originally had purchased it, or if they had sort of spontaneously started it.
Understood. Were there more questions in this, or these are just 5 of the many questions that you asked?
5 of the many questions. Yeah, I'll mention a few more later, but we really asked a lot of questions. We even asked people to do their own sort of sensory analysis of like seeing if their starter had floral notes or fruity notes or buttery notes. And that data was really hard to make sense of, I think, just speaking to kind of, the participant-based aspect of not having a trained nose. But we did do some trained sensory work, which I'll get to in a little while.
Good. Excellent.
Okay. So the first thing that we did once we got all of the starters was we took a little tiny sample of everyone's starters and we sequenced the DNA in each starter. And as we were talking about before, with what we call amplicon sequencing, this can quickly tell you what types of yeast and bacteria were in everyone's starters. So it's a really nice way to sort of screen all the starters and see, okay, what's the diversity of yeast. And And so what you're looking at here, this figure, each tiny little vertical line represents one sourdough starter. And so you can see that a lot of the sourdough starters, like, 2/3 of them, were really dominated by Saccharomyces. And I don't think that's super surprising, right? Like if you think about, yeast that people bake with, that's a pretty common one.
Mhm.
However, in a lot of the samples, we also found other super cool and interesting yeasts that, previously had been known to be associated with sourdough, but in a less sort of comprehensive way. So Kazakhstania is a really nice one, Wickerhamomyces. And the cool thing is if you look at these images, you can just see like how incredibly different the morphology of the actual cells are. And then you can start to imagine, how different the metabolisms are too. And then you can start to think about how that already is really impacting what type of bread you have, right?
Wow. Never thought about that.
Yeah. So this is a parallel graphic, and this is the same thing as before, except now we're looking at the lactic acid bacteria instead of the yeast. And so I'll just point out a couple of my favorites: Fructolactobacillus sanfrancisensis, which we can guess where that namesake comes from. So all of the starters that had that are in pink here. So you can tell it was a bunch of starters. And one of the interesting things is when it was present, it was pretty much the only thing found in starters, right? Like, so this pink line goes from the bottom to the top, meaning it was like the only species found, versus Lactobacillus brevis is this red bar represented across a lot of different samples. Like over 50% of samples have Lactobacillus brevis in their starter. But the interesting thing is, if you have brevis, it looks like, it's found with a ton of other different lactic acid bacteria. And I say this just to point out that, we found over 70 species of lactic acid bacteria in starters, despite, only really a handful that you can find commercially. So there's this huge untapped resource of diverse lactic acid bacteria that, can make totally different sourdough starters that we just haven't captured.
Gotcha. I just out of curiosity, you mentioned earlier that there were projects that dealt with, for instance, cheese, right? Do you know if the same kind of findings were found with cheese as you're finding with sourdough starters?
Yeah, it's such an interesting question. Definitely a lot of diversity in cheese. And I think that one of the main groupings for like how different cheese microbiomes group together in terms of their species is by like bloomy rind versus wash rind, and sort of those sort of key cheese categories. But I think one of the cheeses are a little bit more controlled. Like you have to be intentional about your inoculation of like what microbes are being added to the cheese. And I think that that's one of the really cool things about sourdough, and also, like fermented vegetables, for example, is that like you leave out the sort of raw materials and then the microbes are recruited versus having to inoculate it. Yeah. So
just another follow-up question. So one of the people we had on very early in this Science of Sourdough project was John Porter from Smog City. And he was explaining to us that depending on what kind of beer he was making, he would order different kinds of yeast and bacteria from the lab. Do you think that the bread and pizza world will ever get to that point where we'll have like a menu?
Absolutely, right?
Like how far away are we? Months or years or decades? How far are we from that? I'm feeling impatient now that I know that.
I know, right? Like, can you imagine all of the crazy, cool varieties of
I actually really
that you could
can. I
make
really
with all
can.
of these combinations?
Yeah.
I would say not that far off, and I definitely know that people are interested in doing it, right? So I hope so, because I think it's totally an underutilized resource, right? And we're not taking advantage of this sort of natural diversity that we've cultivated over thousands of years of making fermented foods. So yeah.
I'm also just wondering, like, I know that everybody has like an aroma wheel, right? And you know, now apparently sourdough has it. We know coffee has it. We know wine has it. We know beer and cheese have it. But you know, one of the things as I show this to people who've never seen stuff like this before, and they ask me questions, and I don't have the answers to them because I don't know that much. I just have a few, you know, pieces of information. But one of the things I'm wondering about, like an aroma wheel like that, is that do those aromas that you find in different strains of bacteria and yeast in the combination, do they translate to actual flavors in bread or not?
Yes.
I know this is not in the slide, but I know that I have you, and
No.
I've been wanting to ask somebody.
It's such a good question, and I'm going to tell you about some metabolite data that we have, plus sensory panel data that we have. So basically, the idea is that, you know, different species and even different strains of microbes have different metabolisms, meaning that they're producing different metabolites, which, you know, you can think of as sort of the flavor formation aspect of sourdough, right? And so absolutely, that will impact your final bread flavor. Do I wish that we had like translated this all the way into bread? Absolutely. We didn't for this study. There have been other studies that have started to look at like flavor formation in the final bread product and how it's tied to particular strains of lactic acid bacteria and yeast, which I think is super cool. Yeah.
Thank you for answering. Thank you. Sorry to sidestep your presentation.
No,
Let's continue. I know we have a lot to cover.
We'll get there.
Good.
So this is just to say, you know, I think sourdough has been underappreciated relative to a lot of other fermented beverages like beer, wine, where we have this crazy diversity of yeasts that we can choose from in fermentation. And it's not due to a lack of natural diversity out there. Okay, so after kind of understanding, you know, okay, what species are in sourdough, the next thing that we wanted to know is, can we actually predict who, as in what yeast and lactic acid bacteria are in a sourdough starter based on all of this information that we gathered? So again, like a key piece of information that we got was just like, where was the sourdough sample shipped from, right? So like the geography of the sample. And then we asked participants all of these other questions about you know, what types of flour they used, how they stored their sourdough, did they have household pets, which might sound like a funny one, but it's been shown that there's actually a signature of like a pet microbiome in your house. So if you take a sample of dust from your home, you can actually predict whether someone has a dog or cat just based on the microbes of that sample, which, like, obviously a much easier way to do that would just be to see if you saw dog or cat fur in the house. But the point is that there are microbes that are associated with animals. Do they actually show up in sourdough? The good news is that is no is the answer to that question, I think. That's good news. But the sort of short take home is that, you know, we collected all these process parameters and they really didn't explain that much variation in composition. There were some, definitely some correlations that we picked up that are in the paper about, you know, what type of flour you feed your sourdough with, for example. So that did matter, but the results were pretty messy. And I think part of that is just when you're asking people to self-report, there isn't really room for nuance. So, you know, for example, if someone's like, well, I usually feed my sourdough starter rye flour, but a couple months ago, I ran out and I did wheat flour for a few weeks. Like you just can't capture that kind of nuance in a survey. So we'd really need a like lab-based experiment, which people you know, are doing to see how those process parameters like impact community. But this was still surprising. So we really thought that geography was going to matter way more than it did, which brings me to kind of like my misconception number 2, which is that, you know, I think it's pretty common in sort of baking lore that geography really matters in terms of what microbes your starter have access to. And we really didn't find that. So I'm obviously picking on San Francisco here, but we didn't detect any strong geographic signal for sort of any of our dominant microbes.
I think they deserve to be picked on because they've been doing victory dances for decades.
I know, right?
Right? The Italians too, while we're talking about it. Just saying.
It's true, I know. So this result was kind of really annoying to a lot of people. And yeah, so our joke was that, you know, our paper should have been called San Francisco, You're Not That Special. But we
Love
didn't.
it. Love it.
We refrained.
Love it.
But really, it's a compliment to sourdough, like to San Francisco bakers, right? Because if they don't have special microbes and they're still making amazing sourdough, then that means that like their technique and all of these other things are really important and they're killing it, right? So
yeah,
ultimately, it's a compliment, I think.
It is. I like it. Good reframe.
There we go. Okay, so the other thing that we're really interested in is like, do certain types of microbes, are they commonly found together? Does that make sense? Like if there's a particular yeast, is it always found with this particular lactic acid bacteria? Because that could be another way that we could start to predict like how sourdough microbiomes, you know, are formed and what species are there. So we did this series of lab experiments. So these are kind of like mini sourdoughs that we made in our lab, and we reconstituted wild starters, and we actually isolated specific microbes, and then we competed them against each other. So we'd like put 2 different types of lactobacillus into a tube together and like check who won. So these are a bunch of experimental details that you don't really care about.
No, I care. That's really cool.
So we mixed these sort of the most dominant yeasts that we found across our starters, pairwise with all of these different lactic acid bacteria. So again, we just took a sampling of some of the most dominant ones, and then we put them together, and then we did all of these transfers to actually mimic actual sourdough, right? Because that's what we want to know about. And then we looked at the results and we saw, okay, who likes to co-occur together and who doesn't. And you can see, so here are the outcomes. So this is a co-occurrence network. But what it means is that like if 2 microbes are connected by a blue line, then they co-occur more often than we'd predict by chance. And if there's a red line, it means that they don't co-occur together in our experiment.
Got it.
And so we found that there are all these sort of interesting biotic interactions between different microbes that kind of shape who the resultant community is.
And what does the word biotic mean?
Biotic. So we talk about abiotic interactions. We're talking about things like, you know, flour, or what type of water to use, well water, or, you know, tap water. We talk about biotic, we're just saying the interactions between different types of microbes. So,
got it.
Yeah. Okay. Yeah.
No, come on, we're on a tight schedule. Sorry. I opened my mouth and I closed it. Sorry about that.
Good. Okay, so this is just to say that it still remains really hard to actually predict what is in your starter unless you live literally sequence or culture it. So it's kind of the Wild West out there. And we can't really tell you at this point, you know, if you do this and this and this, you'll definitely get this microbe. Like we're just not there yet, right? Okay. So this is probably the part that I'm most excited about, which is, you know, we are interested in all of these sort of broad biogeographic patterns of sourdough, but really what we wanted to know as bakers is like, how are these different community types that we see, how does that actually translate into functional properties of sourdough? So here we're actually looking at dough rise rates. So again, these are our sourdough systems, and you can actually see the doughs rising. And then our video cut out, but we actually can translate that rate of dough rise just using software into, you know, a nice measurable thing. And this group of sourdoughs actually had acetic acid bacteria in them. And that was one of our interesting findings, is that if you have acetic acid bacteria, which is kind of this underappreciated group of microbes that we found are actually quite common in sourdough starters, but no one really talks about them. So pretty much if you ask someone like sourdough microbes, what are they? They're going to say yeast and lactic acid bacteria. Well, we found in our survey acetic acid bacteria are also really common. I think they were present in about a fifth of the starters. And when they were there, they just totally depressed leavening rates. And they also gave really specific sensory notes. So this goes back to what you were talking about earlier. So we actually measured all of the metabolites, which are on the next slide, that were associated with different types of starters. And then we had a sensory panel actually translate that into, you know, sensory notes of the starter, which isn't exactly like the flavor of the final product, but definitely contributes to flavor formation.
Got it. Just on a side note, I'm very curious about your definition of leavening, and let me just assume that when you talk about leavening, you're talking about dough rising. And one of the things, the reason I'm asking the question is that one of the kind of disconnects in baking with sourdough compared with commercial yeast is that you expect there to be a connection between the amount that your dough rises and the amount that your bread or pizza rises in the oven. But it's not really the case with sourdough. It's what seems to be much more the case with commercial yeast. Is were you able, or have people tested that same idea?
Yeah, that's super interesting. I mean, for this, definitely not. We only looked at, you know, the rate of dough rise in our very model system, right? And I think that would be fascinating. And that's obviously where we need to go with this research to have, you know, direct relevance. So yeah, I think that's super interesting. Okay, this is a crazy graphic. And I only put it up to say that these are metabolites that we actually measured. So this gets back to saying that the different types of sourdoughs have really different metabolite profiles, and it's easy to imagine how that translates into really different actual flavors of sourdough. So I don't think we need to get into the details here at all, but just to say that yeah, all of these different combinations of lactic acid bacteria and yeast together actually translate into really different flavors, and I think, or sensory notes, I should say. And you can see kind of what some of the dominant sensory notes for sourdough were up here. So not surprisingly, if you had acetic acid bacteria, vinegar was a big sensory note. And then there were other types of sourdoughs that were very strongly like green apple.
So those are just a couple of examples, right? Green apple, fermented sour, yeasty, acetic acid vinegar, and other, right? I'm sure you had many more. Am I wrong?
Yeah, we had some pretty gross ones too. Like you wouldn't want to know that your starter was associated with like sweaty feet in corn chips, right? Yeah. Yeah. Cool. So yeah, I already got to this misconception. So just to point out again, we really found acetic acid bacteria had a huge impact on starters when they were there. They were present in about a fifth of the 500 starters.
Only a fifth.
Yeah, but that's like over 100 starters, right? So yeah. Okay, and then I just have a couple slides on what our lab is working on now. So we have these 500 starters that we sequenced, but now we can actually go back and culture all of the cool microbes that we want to study in more detail from those starters, right? So now what we're doing is we're developing a culture collection of all of the different isolates of different species that are really different that we want to go back and take a closer look at. So I'll just give you one example. One thing that we're really interested in is how strain diversity affects microbiomes. The idea here is just like, even within a single species, you can have a ton of genetic diversity at the strain level. So we're really interested in understanding, does that sort of finer scale of diversity translate into differences in sourdough starters? And actually, a lot of people like in microbiome research care about this. For example, we know that different strains of E. coli can be pathogenic or non-pathogenic. So it's a very relevant question to sort of the microbiome community more broadly. So here we actually went back to a subset of these starters and cultured all of these different acetic acid bacteria from the starters. And then we could set up synthetic experiments in our lab. And one of our cool findings is that even within one species, so here is Acetobacter malorum, so even within just that one species, we get really strongly repeatable differences in pH, which would relate to like the overall acidity of the starter. So this is some of the work that we're really excited about right now. And it's also relevant thinking about strain diversity because microbes are always evolving. And one way that we can think about sort of this diversification is by testing different strains. And so I just point out that, even if you've had a starter for a long time and there's all this talk about the 200-year-old sourdough starter, apparently people are selling really old sourdough starters on Etsy. Those communities are evolving and interacting and aren't necessarily, definitely aren't the same community as they were, a month ago, a year ago, right? So sourdough microbiomes are really dynamic too. And I think another sort of really interesting area that we don't understand a lot about is even just the fluctuations that are due to the evolution of the microbes, even in really short time frames. And I get a lot of questions about I was really happy with my starter, but now it tastes different and I don't know why. And I think that that's such a cool and interesting area to learn more about. So yeah.
Does that make you think that when let's say you inherit a starter from somebody, right? And you end up using that starter and loving that starter, should you have a backup in the refrigerator or something? Or even is your refrigerator feeding your starter with whatever else you're keeping in your refrigerator?
Totally. So one way that you can store your starter pretty well is if you freeze dry it, which I don't know if everyone has access to that. But yeah,
well, what do I need to freeze dry a starter?
A dehydrator would work.
Got it.
Maybe it's still a little bit niche. I'm not sure. Actually,
no.
You could even do it. You could even do it in the oven if you do a low temperature. Mm-hmm.
How low?
I want to say, probably I'm just making this up, but
no.
Just out there 150 to 200 degrees for a long time. But then if you store flakes of freeze-dried starter, you can also store it in the freezer. And we have found that we're able to revive sourdough starter that's been stored in the freezer for 5 years perfectly fine, just in the sourdough matrix. So yeast cells can be somewhat sensitive to freeze and rethaw. So there would be risk of losing the yeast that you really care about. But we found actually pretty good revival rates. So
wow.
Yeah, store the backup. I like it.
Yeah.
Cool. Yeah, that's what I had for you. So
that's pretty amazing. And who are all these fine people in this photo?
Yeah, so here's my lab group at Syracuse University, and some of the work that you saw at the end there about strain diversity was done by our research technician, Hannah Rappaport. And then a lot of what you heard about today was the 500 starter project, and all the folks involved in that project are listed over here. So here's the big we that I keep talking about. Yeah, and then these are our current collaborators.
Wow, that's
and
a lot of people.
And you can see I even included a slice of bread in my lab logo here, but so we really need to start getting all the way to bread, right?
Yeah.
Live up to our logo. So
yeah, perfect. Thank you. This has been great. Cameron, you still there?
I'm here.
Welcome back.
Thank you. That was excellent.
Don't you think?
Yes, I learned a lot. That was super fun.
I had to limit the questions that I wanted to ask because we're almost up to our time and we barely finished. She's like, oh, it's a 20-minute presentation.
I made it, right?
No, that was impressive. That's like a lot of information to explain in a really coherent, easy to understand way, I think.
Awesome.
Excellent job. That was awesome.
Really good.
Yes, I think we had a few questions. I tried to answer some of them, but I think maybe give Angela a shot too. We had some good ones. I really liked the one that asked, David asked, does some bacteria grow, consume sugar/flour at a slower rate than others?
Yeah, that's a great question. And the answer is absolutely yes. Yeah. And even different types of carbohydrates. So we know that some species, for example, utilize maltose really, really well, and others can't. And that definitely plays into the dynamics of what we see sort of the resultant sourdough communities to be. Yeah, it's hugely important. Can the microbe utilize the substrate is the first question, and then the rate of utilization is another huge thing. We did a little bit of work on that I didn't include, but yeah, so much more to be done there.
Yeah, the tip of the iceberg.
Yeah.
Something people ask me all the time is that, do you think we'll get to the point one day where maybe you would use, one combination of yeast and bacteria if you wanted, let's say, more caramelization, right? Because of the way that the yeast or bacteria metabolize the sugars, right? Or the rate at which they eat them.
Yeah, absolutely.
One of the issues that a lot of kind of sourdough newbies have is that, however it is that they're storing their starter, right, or feeding their starter, they're running out of sugars, and so when they most need them for flavor and color, they don't have them, and so there's a lot of pale sourdough pizza out there for people who are learning, Yeah. And I'm not saying it tastes bad, but a lot of what we eat is with our eyes.
Yeah, the aesthetic.
Right?
Just like that nice color.
Yeah, and I blame Instagram for ruining people's pizza experiences because, you can have an awesome pizza out there, but people are looking for like big open crumbs or leopard spotting or whatever it is they're looking for. And even though their pizza's bomb, they're like, yeah, but that guy's has big open crumb structure, right?
You contributed to this culture on Instagram. Huh?
Unfortunately, I have. Something I asked Cameron a while back was, do you think that we could get to a point where we could come up with a starter that hit a certain pH and then wouldn't get any more sour? And he said, well, yeah. And I was like, really? Can we start working on that yesterday, please?
Yeah, totally. And it's so, I mean, yeah, to me, there should be a whole line of different starters that do different things, right?
Right.
Because love the super sour, but then to others, it's such a turn off. So
Such a turnoff, right?
yeah.
Awesome. I've been working with some of the starters that Cameron has been feeding me, and it's really a head scratcher for me because I'm so used to working with sourdough and thinking about feeding schedules and temperatures and watching the starter rise and gauging by the size of the bubbles or the smell. And now I'm just like sprinkling, 6 grams of this and 2 grams of that into my dough and just going, and I'm getting these incredible results. And it's like, wow, it's like I imagine that for people who milked cows every morning before they had breakfast, and suddenly the milkman delivers the milk, you're like, this is just not right.
Right.
A
Totally.
truck
Well,
brings
yeah.
the milk, and you
Yeah,
can
but no one,
you
but who
want
wants to
like
go back to it?
non-fat loaf. What?
Yeah, it's like, but only 1% of the population actually wants to milk the cows now, right? It's like,
Yeah.
It's like, well, once we have, these amazing options, are you really gonna want to try your risk at home
Some people will.
Well, yeah, some
Yeah.
people will.
So did we answer all the questions in the chat? We did. Excellent. Well, thank you everybody for all your help in the background. That was very exciting.
I think real quick,
Justin.
I think I got all of the pertinent links in the chat that everybody can look at. So guys, and there is
Good.
the Angela, that story map that you shared with us, that's basically your presentation, correct? So people, if they want to study that and do a little bit deeper dive on that, it's in the chat that everybody, it's available to look at and reread a few times, a little light reading, I guess.
Justin, we'll include all those links when we put this post this on YouTube.
Yeah, they'll be
Perfect.
down underneath with all the little times that I put in on all the slides, etc. So
beautiful. Well, as we wind down, I want to start by thanking Cameron, who's here every day, every night, every week. Thank you for introducing us to Angela. This has been super cool. I've been reading about her for a long time. I think we met a couple of weeks ago on Zoom, so it was great to finally understand. I mean, it's funny to me, like, you people are like heroes now. Like, when I look at the people who contributed to 500 sourdoughs, I actually recognize some of the names, and I even can match them up with some faces that I've seen on other YouTube videos. And I'm starting to realize there's a pretty awesome community of people that's doing this really important work. So I'm glad to begin to connect the dots. So thank you for that, Angela.
Well, yeah, and likewise, and it's been super fun to meet you and to get to share some of our research. So,
and maybe one day we'll be able to make you a pizza with microbes that Cameron or you have harvested.
Absolutely, that's the dream. Right?
I don't know. Nobody really knows yet, but here's some beautiful bread that I baked this weekend, and this is like a little bit left. I'm gonna make my wife a sandwich for dinner. This is gonna be a huge sandwich, so pretty fun actually. It makes pretty delicious bread. I did 3 tastings this weekend, and compared to my first weekend where the microbes hadn't really aligned yet, this weekend I got all thumbs up from some pretty big chefs. So good job, Cameron and James. Very exciting.
Yeah, thanks so much, Angela, for coming. This is awesome, and I love that we get to try and translate some of this stuff to bakers and the taxpayers that pay for all of our research. So it's so nice to be able to try and explain it outside of like our scientific conference. So, and I appreciate you doing that. I know it's like, it's a Monday night, and I appreciate you being here. It was really awesome.
Yeah, of course. And I love all the questions, Noel. So thank you. It's like
my pleasure.
But so fun to actually think about those things, right?
Yeah, thank you. And we included lots of links in the chat. But for people who are watching this on YouTube, I'm just curious if they want to follow your progress. Do you post on your website or on Twitter or X? How do we keep up with you and your work?
I'm on X for now, at least.
For now, at least.
And
no Instagram account, right?
Too. What's that?
No Instagram account, right?
I have an Instagram account, but it's only pictures of my son, not of my sourdough.
All right.
But
Well,
maybe
let us know when you
that's
have an
in the
Instagram for
future.
sourdough. Nothing against your son.
That's right. I know. Yeah.
Beautiful. Great. Well, thank you again. We appreciate your time and all of your beautiful knowledge. Thank you for sharing with us.
Yeah, absolutely.
And thanks to James and Cameron. Justin, I'll throw it back to you. Thank you, sir, for joining us from your vacation.
Absolutely. You know, this is, I like to say it's delicious research, right? All this stuff. I'm so grateful. Angela, thank you so much for coming and hanging out with us. Cam, Jimmy, down there, Sandezi Way. Thank you guys so very much. I'm excited. You know, I haven't had a chance. Noel keeps trying with all these funky chefs, and come on, you gotta kick your boy a little bit of that research, that delicious research. Noel, as always, thank you. Guys, thanks for joining us on this sourdough adventure. I mean, the end of 2023-ish, as we go into Thanksgiving. Thanks for spending some QT. That's always that quality time with Noel and I and the rest of the gang. You know, these are crazy times. So as I always say, please, 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 stay safe, stay strong, belong with one another, the opposite of being short, because we all belong here together. Keep making, keep baking, please, please, please. Guys, you know, we do not get into the politics of pepperoni and pineapple here at Slow Rise. Oh, no. We say forever and ever, pizza always, and always pizza. Pieces and slices, everybody. With love and happy Thanksgiving. Mwah.
Thank you, Justin. Thank you, everybody. Safe weekend, everybody.